All right. How's it going, 10th grade? We are starting in chapter 11. I actually am doing this video again because I can't merge the two videos together using the Zoom recording feature. So I'm doing the whole chapter from start to finish right now. What you probably have seen previously was just from section 1. Okay, so I'll go ahead and share my screen here, and we'll get started looking at the lecture. Okay, so again, chapter 11 is molecular genetics. So this is genetics, not necessarily about the alleles and the chromosomes and how they separate, but rather the fact that DNA is made up of molecules that make this whole thing possible. So obviously, Gregor Mendel had done a lot of work with pea plants and had a somewhat decent understanding of how information got passed from generation to generation, or heredity, the study of how that stuff gets passed from one generation to the next, but he didn't necessarily know why or the specifics of how. So it wasn't until much later that scientists started to try and better understand this. The first scientist that really tried to focus in on the molecule itself that's responsible for heredity was a scientist by the name of Griffith. It was back in the early 1900s, 1928. He was studying the effect of two different strains of Staphylococcus, or Streptococcus pneumoniae, which is a type of bacteria that causes pneumonia. So what he did in his experiment was he took a rough strain, non-virulent, okay, so not the bad one, the one that doesn't cause a lot of harm, that one doesn't cause harm, and then a smooth strain, which was virulent, and he injected mice. And what he found was that the rough strain did not kill the mouse. Okay, obviously he did this lots of times. The smooth strain he injected the mouse with did kill the mouse. Okay, so we can expect that in the future, anything injected with a smooth strain, with a live bacteria of the smooth strain, it's going to kill the mouse. Live bacteria of the rough strain, not going to kill the mouse. Okay, so we know that the smooth strain is going to kill the mouse. So what he did then was he took the bacteria and he killed it using heat, injected it into the mouse again, and look what happened. The mouse lived that time. Well, that makes sense, because when you heat kill the bacteria, the body will recognize, whatever animal it is will recognize the bacteria and then mount an immune response to kill the bacteria. Well, the bacteria is already dead. So yeah, the body will recognize it and will start to attack it, but it's already dead. Okay, so it's not causing too much of a problem. If it were a live bacteria, when that bacteria enters the host, the mouse or the human or whatever it happens to be, it takes over the functioning or the normal functioning of those cells and tricks the cell into making DNA for the bacteria. Okay, that's why we end up with bacterial infections and things like that. So what happened then, he took this heat-killed, smooth one, injected the mouse, and the mouse lived. Okay, that makes sense because the bacteria was already dead. Okay, so then he took more of the heat-killed bacteria, mixed it with some of the smooth strain, or the rough strain bacteria. Okay, look over here, rough strain. And the rough strain doesn't kill the mouse. And the heat-killed smooth strain, that doesn't kill the mouse. So we'd expect that when you mix those two together, it's not going to kill the mouse, but that's not actually what happened. The mouse was injected and ended up that the mouse died. Okay, so this has Griffith thinking that there's got to be something going on there. The heat-killed bacteria must be having some sort of an effect on the smooth strain, because previously the smooth strain didn't kill anything, and heat-killed isn't going to kill anything. Anyways, okay, so he's starting to wonder now what's causing the mouse to die, and this prompted more investigation by other scientists. What was actually going on there? What was the molecule? So Hershey and Chase in the 1950s were working on more definitive proof of what was actually causing the transformation of those bacteria, to the point where the bacteria that were still alive all of a sudden gained the ability to be able to infect the host. whereas before it didn't. So they started doing experiments using bacteriophages. Those are viruses that will attack bacteria. Okay, a bacteriophage is made up of protein and DNA, and most viruses, they can't reproduce on their own. They have to have a host cell of some sort and able to reproduce. So what they did was they used a process of radioactive labeling, and they labeled different parts, radioactively labeled different parts of the virus, the DNA part, and then, well, here, let me back up a second. You can trace the DNA and the protein as the bacteriophage infects the bacteria, because it's going to start reproducing. And if it starts reproducing radioactively labeled bacteria, that means that it was producing radioactive molecules. So one group was labeled with bacteriophages that have radioactive phosphorus. Okay, phosphorus is not something that gets used in proteins, and one group used radioactive sulfur. Sulfur is not used in DNA. Okay, so phosphorus gets used in DNA, and sulfur does not. Okay, this is important to try and keep that in mind. All right, so here's what he did. He took these bacteriophages and they infected the bacteria, and what happens is the DNA, or the genetic material, gets into or injected into the bacteria cell. Okay, so this is the new virus DNA right here, virus DNA. Okay, now other parts of the virus are protein, and so after the bacteriophage infects the bacteria, what they found was that the cell that had the radioactive phosphorus, okay, which was part of the DNA, was responsible or being transferred from one bacterial cell to the next. Okay, so then they were able to conclude that the genetic material that's getting passed from one generation to the next is in the nucleus, it is the DNA. Okay, the DNA is being passed from parental generations to offspring, and that's how, giving us a much better idea of how the genetic material gets passed from one generation to the next. Okay, so in the 1920s, other scientists they're doing research at the same time, and they figured out that the DNA itself has a structure. It's made up of these 5-carbon ribose sugars, a phosphate group. Here's your phosphate group right here, and then the nitrogen group. Okay, now the nitrogen groups can be a little bit different. They aren't exactly the same. We have adenine, guanine, cytosine, and thymine. And if we're talking about RNA, which we will in the next section, it uses uracil. So we'll get into the details of that in a minute. So these nucleotides, or these groups, are connected together and they make these big long chains. Okay. So the nitrogen bases, adenine, guanine, thymine, and cytosine, and then if we're talking about RNA, we're talking uracil as well. They together in these big long strands create the DNA strand. Okay. All right, so another scientist by the name of Chargaff was studying the relationship between these different nitrogen bases, and what he found was that depending on the type of organism he was studying, and it was specific to every species, the amount of adenine was exactly the same as the amount of thymine, and the amount of guanine is exactly the same as the amount of cytosine. Okay, so he came up with what's called Chargaff's rule. Adenines, or the amount of adenine, is equal to the amount of thymine or cytosine, and guanine is exactly the same amount. So they come in a 1-to-1 ratio. For every adenine, there has to be a thymine. Well, this helped them to come to the conclusion that, okay, these things must be paired up then. Because if we have exactly one of each, A's are pairing with T's, then we would have just as much thymine as we do adenine, and that's the way it works out. Okay, guanine and cytosine, the same thing, exact amounts. So this, with Chargaff's rule, and knowing now that these nitrogen bases are pairing up is helping other scientists figure out what the structure, the actual physical structure of the DNA molecule is. Okay, so they used Chargaff's information, and I should back up a second. Rosalind Franklin, Maurice Wilkins, Watson, Francis Crick, and James Watson were the scientists that were working really hard to try and figure out the structure of DNA. Okay, now they were doing different stuff, but probably one of the more helpful things was that Rosalind Franklin was using a process called X-ray crystallography, where basically she was taking X-ray images of DNA molecules, and she came up with what we call the famous Photo 51. Okay, we'll talk about Photo 51 in a little bit here. It's hard to look at that picture or that image and think to myself, oh, well, that creates the structure that we see over here on the right. This is what we call a double helix. You know, if you're a physicist, this makes a heck of a lot more sense than it would for just a regular person trying to understand the structure of DNA. Okay. So Watson and Crick now, they're together using Chargaff's information, and we see their A's are paired with T's and C's are paired with G's, and so they come in equal amounts, and they basically they used Rosalind Franklin's Photo 51, which we'll get to in a little bit more detail in just a second here, along with Chargaff's data to create a model of what they think it might look like. And what they came up with was this double helix. Their research and what they determined that it looked like, or its actual structure, matched up with everybody else's research. And so they proposed that the structure of DNA was this double helix, which is like a big twisted ladder that had nitrogen bases or base pairs in the middle that are complementary to one another, meaning that the shape or the molecule, they complement one another because they're able to link up or to bond with one another, if you will, through hydrogen bonding. Okay, this also confirmed Chargaff's rule. Now, it was a theory, and it actually turns out that his idea about the fact that they would come in equal parts works out pretty good, because every time you have an adenine, there's a thymine connected to it or bonded to it. Every time you have a guanine, there's a cytosine. Okay, so A's and T's pair, and G's and C's pair. And it's going to be really important that you can remember that, and we'll go into more detail of that in different section. Okay, so the way that the two strands are oriented is what we call the 5' or the 3' orientation. And it just has to do with what side of the twisted ladder that we're talking about, okay? So carbon atoms are labeled in 1 through 5. So if you look, there's carbon number 1, number 2, number 3, number 4, and then there's carbon number 5. It's carbon number 5, that's the 5', and carbon number 3, that's the 3'. Okay, so the 3' end attaches to the 5' end. Okay, so there's your 3' and there's your 5', and they are connected through that phosphate group. Okay, these are what we call anti-parallel strands. That means that they are running opposite of one another. So all of this information was provided enough evidence where Watson and Crick could actually say that, hey, this is the structure of DNA, this is what it's like, and we can move forward from there. You know, Wilkins and Franklin, they published articles as well, but Watson and Crick are the ones that got most of the credit for it because they came out with it first. Now, in science, yeah, we're supposed to all be nice and sharing information, but there's a lot of competition amongst researchers in the scientific fields. And what ended up happening is that Watson and Crick somehow got a hold of Rosalind Franklin's Photo 51. They basically stole her information and used her information to come up with the structure of DNA. Now, a lot of other people were working on this at the same time, but it's because they were able to get their hands on, or that they stole, if you will, her Photo 51. They were able to come up with the structure. They published a paper first. They were given credit for it. They won a Nobel Prize, which in the scientific world, that's, you know, it's a really big deal. Rosalind Franklin subsequently ended up dying from cancer, which could be expected because she was working with X-rays. And at that time, in the 1950s, scientists and researchers didn't know that X-rays were that damaging or could be causing that many problems. Rosalind Franklin ended up passing away from cancer, and she was given the Nobel Prize years after her death. So she still is given credit. It's just that Watson and Crick get most of the credit. Here's a picture of Watson and Crick and the model that they made. So now we have a better understanding of what the actual molecules are like. And together, those make big, long chains of big, long chains that eventually turn into chromosomes. A human chromosome can be anywhere from 51 to 245 million base pairs, okay? If we had a strand that was 140 million base pairs long, it would be about 5 centimeters, okay? So, you know, about that much, if it were laid out in a straight line, okay? The DNA then, it gets coiled around these histones. So the DNA has these long strands, gets coiled around these histones, and together, those create what we call a nucleosome, okay? That's that wrap of DNA and protein histones. And they together will make these chromatin fibers, okay? We talk about when we have chromosomes that decondense or uncondense when they're going through prophase of the cell cycle. So we can see this, what we're looking at here with the histones and the nucleosomes. These structures are what we end up with once everything is uncondensed inside the nucleus. Now, when they condense, they turn into chromosomes, which is good. They condense during prophase, and then they uncondense after mitosis or meiosis is finished. And that's important because otherwise it would be very difficult for DNA to be able to be replicated. Okay, that was it for 11.1. I'll take a quick pause for a second here, and then we'll go on with 11.2, which is how DNA is able to be replicated. Okay, this is a pretty short section. DNA replication happens in, it's what we call semi-conservative replication. That means that one strand gets used as a template, okay? So here's the complementary strand that's, you know, complementing this one. We'll go into a little bit more of the details of this, but what happens is that the parent strand will remain in the original nucleus, and the other one will get taken out and over to the ribosome so that it can be used as a template for making more proteins. Okay, so one strand, it's semi-conservative. So as those strands separate, the parent strand remains behind and gets used as a template, and the new strand then is taken elsewhere. Okay, so here's how the whole process works. There's a protein, or an enzyme called DNA helicase. We can see it right here. This protein unwinds or unzips, if you will, the double helix, okay? Hydrogen bonds that hold those nitrogen bases together, hydrogen bonds are broken, and they're able to separate then, and it leaves behind these single strands of DNA, okay? Now, there's single-stranded binding proteins then that help them to remain apart so that the process of DNA replication can happen, okay? So RNA polymerase is adding these short little segments of RNA on each strand. So DNA helicase here is doing the unwinding. DNA polymerase then is adding nucleotides, or those nitrogen bases, adenine, guanine, thymine, cytosine. It's adding those nitrogen or those nucleotides to the new DNA strand, okay? And it creates what we call Okazaki fragments, okay? Now, there's two different replicating strands. The leading strand, if you look at it over here, is going towards the replication fork. And the replication fork is, you know, like a strand of DNA that's constantly being split. And so it's opening up further and further and further. So we can add to this strand very easily, and it just keeps going because it keeps opening up, opening up, opening up, opening up. Okazaki fragments happen on the lagging strand because they open up and then they eventually have to close back up again. Okay. So the lagging strand replicates away from the replication fork, and that creates a situation where it's like leapfrog. You can work ahead a little bit and then you have to stop and go back because it has to close back in on itself. The strand of DNA has to close back in on itself. It's not just open forever. Okay. So they are connected by DNA ligase. Each of those little sections is 100 to 200 nucleotides long. Okay. And those are Okazaki fragments. I promise you'll see that again. Okay. So there's some significant differences between bacteria versus plant animal cells, or eukaryotic versus prokaryotic cells. The process in plants and animals, or eukaryotic cells, that's cells that have a nucleus, happens the way that we just explained and just went through. Okay, it happens in multiple locations all at the same time. So you have you might have a strand of DNA that, let's just say it's 10 feet long, and it's, the replication is happening in multiple locations all at the same time. Prokaryotic DNA is a little bit different. Eukaryotic DNA is in a double helix DNA strand. Prokaryotic DNA is also double helix, but it creates a circle. Okay. So prokaryotic is circular. Bacterial DNA is in circular strands, and it replicates at two different locations at the same time. It starts at the origin of replication, and it starts here, it works its way around the circle, and then it stops. Okay. And it goes in both directions. Okay, so that was it for 11.2. I'll pause for just a second, and then we'll go on with 11.3, which is about DNA, RNA, and protein. Okay, 11.3 is about DNA, RNA, and protein. This is something that we in biology, we call the central dogma, or it's kind of like the main idea of biology. Okay, DNA makes RNA, RNA makes protein, and protein essentially does everything for living things. Now, obviously, we need a lot of other stuff too, but protein is mostly everything that humans need, or the majority of everything. So here we go. DNA is the instructions for making proteins. Okay, these proteins are like the building blocks. Sometimes we think of it as like a blueprint. Proteins do almost everything for living things. Okay, like if a builder was going to build a house that they had never built before, or maybe they have built it before, they need to use a blueprint. There has to be some instructions to make sure that they do everything along the right timeline and when they need to do it, and that it's done appropriately. Otherwise, you end up with things being messed up. Okay, so DNA makes RNA. Okay, DNA is the code or the blueprints for making RNA. Okay, RNA is guiding the synthesis or telling the ribosome how to make the protein. It's like the instructions, like a recipe for making the proteins. And the proteins then do almost everything for living things. So RNA is ribonucleic acid. It's kind of similar to DNA. DNA is deoxyribose nucleic acid. It contains a sugar ribose, or a ribose sugar, nitrogen bases. But in the nitrogen bases, we talked about the A's, the T's, the G's, and C's, adenine, thymine, guanine, and cytosine. Well, thiamine gets replaced by uracil. Okay, so adenine, instead of pairing with thiamine, it would be uracil pairing with thiamine, and when we're talking about RNA. Okay, there are three different types of RNA. There's messenger RNA, ribosomal RNA, and transfer RNA, or mRNA, rRNA, and tRNA. So messenger RNA is the first one we'll talk about. mRNA, these are long strands of RNA that are complementary to the strand of DNA. Okay, so when we talk about the unwinding, DNA helicase is doing the unwinding, and then this parent strand okay, is going to remain in the nucleus. But as that opens up, mRNA, RNA polymerase, I should say, adds these little nucleotides, the adenines and the uracils and the guanines and the cytosines, to a strand of mRNA, okay? So that strand of mRNA comes in, it gets made complementary to the DNA strand, all right? The mRNA then goes from the nucleus of the cell to the ribosome in the cell, and it tells the ribosome how to make certain proteins, okay? Now, ribosomal RNA, or rRNA, is associated with proteins that form ribosomes in the cytoplasm, okay? So basically, they're just there to help make ribosomes. Now, tRNA is a little bit different. tRNA makes smaller segments that transport different amino acids to the ribosome. Now, we're going to learn more about these amino acids a little bit later on, but the arrangement or how these amino acids are put together in long chains determine the type of protein that something is going to be. So the first step then is transcription. That happens when we have mRNA that gets made from DNA. Now, if you think about transcription, it's like a copy of something. You know, back in the day, a transcriptionist might be somebody that worked for a doctor's office, and the doctor would dictate, or they would talk to a microphone, and then a transcriptionist would write down or put into a document form everything that the doctor said. That way the doctor doesn't have to spend time typing things and in a medical record. So that's what transcription is. It's basically a copy, a complementary copy of mRNA that gets made from the original DNA strand, okay? So that code from the DNA gets transferred to the mRNA, and this happens in the nucleus. The mRNA then, okay, here's the complementary mRNA, gets taken to the cytoplasm, where it's going to end up going to make more proteins in the ribosome. Okay, so what happens is RNA polymerase, that's the enzyme that is this big green enzyme, might be a little tough to see. This big green enzyme is responsible for regulating how that RNA gets made. Okay, so RNA moves along the DNA strand. Okay, this red strand right here is the RNA. And as it's going, it's adding a uracil, an adenine, and there's a thymine there. So another adenine would get added on here. Here's a cytosine, so there's going to be a guanine. Okay, you can see that right there. So this strand is being made complementary to the other strand. As it goes on, it adds what we call codons to the mRNA strand. Okay, the codon is the each three sections right here. So like CAU, that's a codon. UCG, that's a codon. Okay, so there are also what are called introns. Those are the coding sequences. They aren't in the final mRNA strand. The exons are coding sequences that are in the mRNA strand. Okay, so you can see the introns and the extrons here. Introns, they don't get used. The exons, they do get used. The introns get removed before the mRNA leaves the nucleus. Okay, so the code itself, so people ask or talk about the DNA code, this is what they're talking about, okay? So the codon is each of these three bases, okay? So let's just look at this one. Methionine is a start codon, adenine, uracil, guanine, okay? That's the three amino acid, or that's the sequence of those bases that makes up that codon. And that codon is the letters, and it actually codes for methionine, which is the start codon. Basically what that means is that it tells the DNA strand, like, hey, that's where we're going to start making, when we make the new protein, that's where we're starting, okay? So it starts with the amino acid methionine. Now, there are 20 different amino acids, and depending on the order of the amino acids, like it goes proline, and then arginine, and then lysine, and threonine, and then alanine, and valine. Okay, those all go in order. And depending on the order that those amino acids are placed, determines the type of protein or what function that protein is going to have. Okay, and so it goes all the way back down to that base pair level. The adenines, guanines, thymines, and cytosines, or ultimately uracil, because we're talking about RNA now, that determines what the protein is actually going to be. So the process of translation then is happening when the code that's been sent gets read and translated to make the protein, right? So mRNA is made in the nucleus, it's transported, it's taken to the ribosome, and then the ribosome then makes the protein. Transfer RNA acts as what we would think of as like the interpreter, okay, of the mRNA sequence. This big upside down looking T, this is the tRNA. Okay, so tRNA enters the ribosome, anticodons read the code, and it adds amino acids to what we call polypeptide chain, okay? So the chain that gets created is called a polypeptide, okay? It continues until a stop codon gets reached, okay? We talked about those amino acids in the previous slide. When we reach a stop codon, then it tells it to stop, and then you're done. The protein has been made. Okay, so here's your long chain of polypeptides. These are all proteins, or a chain of amino acids that's going to make a protein. That protein gets released and it goes to do whatever it is that it needs to do. Okay, we'll pause here for just a minute and then we'll go on with 11.4, which is gene regulation and mutations. Okay, 11.4 is gene regulation and mutations. And yes, what you're looking at here is a duck that has 4 legs. This is actually a real thing. If you ever take a developmental biology course in college, you'll learn about Hox genes, and you can take undifferentiated cells and place them in different locations, and you can get things like extra legs to grow where a wing should have been. Okay, so gene regulation happens because the organism has some control about the genes and which genes are transcribed in response to changes or stimulus in the environment. Okay, so they started doing studies with this with rabbits, right? They shave a rabbit, put a cold pack on its back, and then all of a sudden we get black hair growing. Well, the black hair is growing because the proteins to make the hair are different now because the environment has induced or told the DNA, like, hey, we're going to start making proteins for hair, except they're going to be a little bit different because they need to be darkly colored from now on. Okay. So a mutation then is a permanent change in a cell's DNA. Cells get mutated all the time. Most of the time, they're not a big deal. Those mutations aren't a big deal. Those organisms are able to overcome that pretty easily, but sometimes they aren't. And if they aren't, then it becomes a permanent change that isn't able to be fixed. Now, there are different mechanisms inside of animals that help to fix damaged DNA. But once it gets damaged to a point, it can't fix itself. And it creates these mutations. There are a few different types of mutations. The first one we'll talk about is called a point mutation. And that happens when there's a change in only one base pair, okay? So we have these bases, right? And let's say we have CAT, 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 and they put it together repetitiously because it's a little bit easier to see. Now, CAT is the codon that codes for histidine, okay? And we would be able to determine that by looking at, okay, we would look at the DNA code, and we find the C and A. Well, actually, CAT doesn't exist because it would need to have the uracil. Okay, but this is where we would go to look and find out what type of protein or amino acid is being made. Okay. Okay, so here's the original sequence. Okay, and we can see that CAT, 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 C, this is CCT instead of CAT. So what's happened here in the original amino acid sequence, histidine is in there, But now, since it's not CAT, it's CCT, codes for proline, okay? So we've screwed up the chain of amino acid, and that's gonna make a different protein, okay? And we call it a point mutation because it's one specific point where something has happened. Now, this is what we call substitution. One base is exchanged for another one. So in this case, we have C or cytosine replacing adenine. And that's just one single base, okay? It gets exchanged for the other. Now, if it's just altered so that it makes a different amino acid, then we call it a missense mutation, okay? Or a missense substitution mutation. A nonsense mutation would be like this one over here. We talked a little bit ago about start and stop codons, or the amino acids that code for to start and stop. So if we look at it, okay, CAG, 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 and then all of a sudden we get to here, it's CAG, but in the corresponding or the complementary strand, it's not CAG, it's the C gets replaced for a T, okay? The cytosine gets exchanged for a thiamine, okay? And that's causing a mutation. And that mutation codes for, or is the codon for, a stop protein, or a stop codon. That stop codon is going to tell everything to just stop happening. And of course, you're going to have problems because that's a protein that is not going to be made, that isn't going to be able to be made correctly, okay? If that's happening in a cell, that can lead to all sorts of other problems. Now, we also have what are called frame shift mutations. These are what we call insertions or deletions, because an added nucleotide gets into the DNA sequence and it bumps everything down a little bit, okay? So if we look at this again, we're looking at the same sequence, and this is causing a frame shift mutation. So we have CAT, 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 so forth and so on, CAT all the way down the line, and it's coding for histidine, okay? Well, the mutated sequence now, we have CAT, 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 and now instead of CAT, we have an insertion of an adenine, okay? Oh, let me back up. That adenine isn't replacing something else that was already there. It's just being added to it, right? So we still have CAT, 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 but the problem is that we've shifted the frame of codons, right? So the three letter sequences have shifted. So we have now, instead of it being CAT, 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 it's ACA. Well, that screws up the next frame of 3. So then it's, where did my cursor go? Okay, so ACA, and it screws up the frames that are coming down the, down the line here, if you will. Okay, now we have ACA, TCA, TCA, TCA. Well, TCA codes for cyranine, not histidine like it's supposed to, okay? So first, ACA would code for threonine, and then after that, because we don't have this insertion the rest of the way down the chain here, or through the sequence, now it's going to be serine, 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 and that's not good, okay? So that's an insertion. A deletion has kind of the same problem as that CAT, 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 okay, so we notice it down here, CAT, 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 C, but the A gets deleted out. So then we have, instead of CAT, we have CTC, which codes for leucine, okay? And now the permanent sequence going from there is that 3-letter sequence, ATC, 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 and it'll be ATC the rest of the chain, okay? It creates, when that happens, it creates the wrong amino acid sequence, which creates the wrong proteins, okay? Those are called frame shift mutations. Now, lots of things cause frame shift mutations, and we call them mutagens. Sometimes we call them carcinogens. There are certain chemicals in our environment that can mutate DNA. radiation, whether it's UV radiation from the sun or tanning beds, or maybe, you know, X-rays or gamma rays. Those are other types of electromagnetic radiation. There are also things called free radicals and ultraviolet radiation. All of those things are mutating DNA in living things. Okay. Now, hopefully our body has mechanisms that can help to fix that, but if it can't, you end up with things like cancer. Okay. So skin cancer, you know, obviously ultraviolet radiation from the sun can mutate the DNA in your skin. You can end up with skin cancer, can be caused from a few different things, but we typically associate skin cancer with having too much access or too much time in the sun, or like tanning beds and things like that. Usually what happens is we end up with these skin blemishes that we call melanomas. And if they have, you know, asymmetry, meaning that they're nice, not sort of the same shape all the way around, or they have irregular borders, or they're weird colors and things like that, or if they're large, there's a higher likelihood of them being cancerous. Okay, so I know that this is a tough thing for you guys to learn when you're not in the classroom. Do your best. If you have questions, please reach out to me, shoot me an email, phone call, you know, whatever we need to do to make to help you guys learn this stuff. Okay, so until next time, take care, stay healthy.